Finding microsatellite loci and making primers for them in Galearis rotundifolia. There is currently a global biodiversity crisis of immense magnitude. Approximately 1 million species faced extinction risk in 2019 due to human-caused destruction of the environment, and such destruction has only increased since then. Humans are using more resources than the Earth can regenerate, and this is leading to the Earth not being able to use its regenerative capacity to sustain other species. This is causing us to enter Earth's sixth mass extinction event. This is having great impacts on humans and human society, and the impacts will only increase as time goes on. This is because the Earth's ecosystems need their biodiversity in order to continue providing us with the vital ecosystem services we rely on to live. These services include giving us oxygen, fertile soil, clean water, protection from disasters, and more. Therefore, human beings have a vested personal interest in safeguarding biodiversity. Not to mention that every species has an inherent right to exist just as much as humanity has an inherent right to exist, and the loss of any species is an immense tragedy in and of itself and should be thought of as such. Indigenous peoples all over the world know this, and they feel kinship and connection to the other species. They believe that humans should be in cooperation with nature instead of being in conflict with it. Plants especially play a vital role in the ecosystems they are a part of. They produce oxygen, which many organisms need to live. They also produce food for many animals. Different animals need to eat different plants. Plants, including orchids, are often in symbiotic relationships with fungi and help the fungi to do their vital job of recycling nutrients into the soil. They help decrease erosion, and they do many other jobs in the ecosystems they are a part of. The Wagner Natural Area is a forest and wetland ecosystem near Edmonton. It is made of marl ponds fed by underground springs, which are surrounded by forests with trees such as spruce, aspen, birch, tamarack, and willow. There are many species in this area, including rare carnivorous plants, and 16 of the 24 orchid species found in Alberta. Galearis rotundifolia, or the roundleaf orchid, is a terrestrial orchid found throughout northern North America, stretching from the North United States through Canada and even in Alaska. They have white or pink flowers with purple spots, and they have a round leaf at their base. They grow in alkaline wetlands. While these flowers are not at risk of extinction for now in the northern parts of their range, they are at risk in the United States, with many American populations having been extirpated. Researchers look at microsatellites to assess the genetic diversity of a population. Populations with more genetic diversity are healthier and can adapt better to change. Genetic diversity decreases when species decline, so changes in genetic diversity can tell us the vulnerability of a species. Microsatellites can tell us if there is inbreeding in a population, which is unhealthy. Microsatellites can also tell us about connectivity. More connected populations are less likely to lose their genetic diversity. If a species has less connectivity than it should, that means its habitat has likely been fragmented. For example, if two close populations living in the same environmental conditions with no natural geographic barriers between them are not genetically connected, there is likely human-caused habitat fragmentation going on. However, sometimes populations don't interbreed because they live in and are adapted to different environments. Seeing whether different environments are genetically connected tells us which population should be kept separate in conservation efforts such as breeding, relocation, and reintroduction. Microsatellites have many uses in informing and guiding the conservation of plants. Knowledge learned from microsatellites can inform management strategies, for example, by helping us choose populations of management priority. In our research, we are going to develop microsatellite primers for use in Galearis rotundifolia. First, we are going to take leaf tissue from different Galearis rotundifolia plants in the Wagner Natural Area. We are going to extract DNA from this leaf tissue. We are going to do this by isolating the DNA and then fragmenting it using restriction enzymes. We will ligate adapters onto the DNA fragments. These adapters will be known sequences of DNA that will be added to each side of each fragment. Next, we will PCR amplify the DNA fragments using primers complementary to the adapters. This way, we won't need to know anything about the fragments we're amplifying. We can just have primers for the adapters. The amplification products, which have many fragments of DNA from many different parts of the genome, will be enriched for microsatellite containing fragments. The way we will do this is by putting DNA probes into the solution. These probes will have base pairs repeating again and again in a pattern, much like the microsatellites do. They will therefore be able to bind to microsatellites in the orchid DNA. The probes also have a streptavidin-coated metal bead on each of them, and these beads can be pulled down with a magnet, which would also pull down the probes and the DNA fragments they're hybridized to, the majority of which contain microsatellites. We will take those beads and release the DNA fragments and then do next generation sequencing on those fragments to get their sequences. This will give us a sequence file of millions of base pairs. We will use the software Genius to look at all the sequences and to find microsatellites within them. Primer 3 will help us to create primers for the microsatellites we find. We will look for microsatellites whose primers have very similar annealing temperatures to the other microsatellites we're developing primers for. So all the primers for all the microsatellites we develop should have very similar annealing temperatures. We are going to then figure out what the optimal conditions are for doing PCR with all of our primers. We are next going to do multiplex PCR to amplify all the microsatellites we developed so that we can test them out. Multiplex PCR is when you amplify multiple targets at the same time using multiple sets of primers. Each primer pair will have a differently colored fluorescent label. Therefore, each microsatellite locus that is amplified will have its amplified fragments fluoresce a different color. We are going to run the amplified products on a capillary gel and look at how many bands are each color and what position those bands are in to see how many microsatellite loci are heterozygous and what size the alleles for each locus are. All the information about the methods is from Dr. David McFadden. Having primers for microsatellite loci in Galearis rotundifolia will allow future researchers to quickly and easily monitor the genetic diversity, inbreeding, vulnerability, genetic structure, and reproductive modes of the species. This will help inform and guide management, for example, by figuring out if any population is declining, by figuring out the number and distribution of management units, and figuring out which populations are of conservation concern or priority. The primers we will make will all work in the same PCR conditions, which makes it even easier and faster for future researchers because they will be able to look at the multiple microsatellite loci for one individual using only one PCR reaction. It is important that we know the health, including genetic health, of as many species as we can, because every species is interconnected with every other species in the web of life. The health of every species affects the health of every other species and the health of the biosphere that all life, including human life, depends on. And these are my references.